My name is Mike. I'm a research associate with the ectoderm team over here at Trailhead Biosystems, and I'm just going to do a quick overview of one of the latest developments we have coming out of our lab, and that is a novel small molecule-based protocol for the directed differentiation of iPSCs into DRD2 positive committed LGE neuroprogenitors. Now, Huntington's disease, obviously, as you may know, has no cure, and it's the gene mutation that causes Huntington's disease that actually leads to that loss of DRD2 cells inside of the forebrain, and that is the reason behind the slew of motor, cognitive, and psychological disorders that characterize this devastating disease. Now, it's this that becomes the impetus that drives the development for the protocol for a DRD2 positive cell population. So what we do here at in the Trailhead pipeline is we employ our high-dimensional design of experiments patented technology in order to develop a small molecule-based, media-based protocol in order to control the cellular fate. Because unlike other companies that engage with transfection or gene editing, we like to keep our process as closely aligned with natural neurogenesis as possible, and we found that we can do that while reducing off-target effects with our media-based protocols. And if I can just talk real quickly about the development of medium-spiny neurons, they initially originate in the LGE region, where then they migrate down into the striatum, where they finally develop into DRD2 and DRD1 subtype MSNs. In the interest of finding a precise population of MSNs to work with here, we assessed the targets of interest through single-cell RNA-seq, and we found the correct region to be the ventral side of the lateral ganglion eminence, which gives us the targets of MEIS2, FOXP1, and PANC, which we've seen develop into DRD2 positive MSNs. So in the beginning of the development of our protocol here, we wanted to ensure that we are committing early on into the LGE region of the brain. So if we can see these figures here through our ICC data, we have a number of markers that we wanted to focus in on, such as OTX2, SIX3, PAX6, and FOXG1. Now, SIX3 and FOXG1 are the interested parties here, where we're going to make sure that we maintain that LGE identity. We also tested for NKX2-1 as well as PAX6, which is going to give us our negative control. That's the medial ganglion eminence as well as the dorsal regions that we want to avoid here. And we can see from our ICC that we have no expression of those two markers. Now, moving on to the next step in our protocol development is the optimization of FOXP1. FOXP1 is an important marker to ensure that we have neuroprogenitors of the LGE lineage, and we can see from our ICC here that we have the FOXP1 in over 80% expression in our population here, as well as SIX3 to make sure that we are also still maintaining that identity in the LGE in general. And we have corroborating data from our PCR that shows we have very high expression of FOXG1 as well. Now, moving on towards our maturation, after we have our LGE committed neuroprogenitors, we want to ensure that we can get these cells to express DRD2. And like I said earlier, Trailhead is committed to ensuring our protocols are as closely aligned with natural neurogenesis as possible. So we have a few markers here that ensure that we are maintaining that process. We have DCX, which is an immature neuronal marker that's an indicator of migrating immature neurons, and we have expression of these as well as they begin to cluster up. We have our CTIP2, which is a post-mitotic neuronal marker, which is over 90% expression, as well as our SIX3, which is proving that we are staying in that LGE region of the forebrain, as well as DRD2 within those migrating clusters. You can see it beginning to express very early on in the neuroprogenitor stage. The next stage in our development is the test of purity. So we performed flow cytometry and found that we had a FOXP1 and CTIP co-expression of 83%, which shows that we do in fact have an 83% population of committed MSN progenitors in our protocol, as well as our 70% of SIX3 showing that we are still maintaining that LGE identity along with 90% expression of FOXG1. Now, just for fun, we decided to move from our adherent protocol to our 3D space, and test the robustness of our differentiation protocol in a small scale bioreactor. And as we can see from these figures here, we were still able to maintain over 83% of co-expression of CTIP and FOXP1. The next step in our process here was to characterize terminally differentiated neurons. We initially tested candidate maturation media and found a very high expression of DRD2 from our PCR readings. And you can see from the figure on the right here, this is the culture that we took our PCR data from. Very beautiful neurospheres connected together by axonal bundles. Now, in the future directions for this protocol is obviously more optimization of maturation media, as well as functional validation through electrophysiology. We want to get into a bunch of different co-culture experimentations with our own Trail Bio astrocytes, as well as A9 dopaminergic neurons. We want to do neurotoxicity assays, and finally our bulk and single cell RNA sequencing. Thank you.